This podcast was produced by OAR FM Dunedin with support from New Zealand On Air.
The Science Notes podcast on OAR FM Dunedin, brought to you by the Division of Sciences at the University of Otago. I'm Dave McMorrin. Over the next half hour, I'll be chatting with a graduate science student about their research, their passion for science, and the things that fuel their creativity. Well, hello, and welcome again to the Science Notes podcast. My name's Dave McMorrin, and this time our guest is Toby Brown. Hi, Toby.
G'day, how you going, Dave?
I'm well. Thank you for coming along
on
this remarkably sunny Dunedin day.
Yeah, it's nice. Bit of a chill in the air, but that's not atypical, I suppose.
No, it's a perfectly normal July day, except that it's almost November, but that's all right. Toby is doing a PhD in biochemistry.
That's right.
At Otago. How far through are we?
So I'm about, I'd say 2.5, 2 2/3 years in, so I'm closing in on the finish line.
Well in,
yeah,
and up to writing.
Yeah, that's right. Yeah,
the
really,
best part.
Really going to crack into it, eh? Yeah.
And so the plan's to be finished by early next year?
That was the original plan. If I'm honest, probably further into next year, but that's fine. That just gives me the time I need to really create the narrative I want with my thesis.
Excellent.
And,
it's all about the story.
Exactly.
Excellent.
Yeah.
Now that you've got the results, now you have to turn it into a story.
That's the one, yeah.
It's best than the other way around.
Yeah.
So how is it that, before we get in, so you, as we're going to be talking about, you've been spending the last 2 2/3 years looking at photosynthesis.
That's right.
And the details thereof.
Yeah. How
is it that you find yourself studying photosynthesis at Otago?
It's a great question. It's not something that I had been thinking about, for years prior. I came into Otago just to do an undergrad. I was into my biochemistry and chemistry, finished that Bachelor of Science, and I decided I wanted to do an honours year. So I had the option in the biochemistry department to look at a few different labs and choose what I would like to do. And Julian Eaton-Rye, who's my now supervisor, seemed incredibly enthusiastic and supportive. And I was immediately engaged with the way he approached science and his passion for photosynthesis. And so I ended up in his lab doing an honours year, and I liked it so much I stuck around.
Right.
Yeah.
So honours, for those of you listening who might not know, is just about, it's a year, but in reality that's about 8 months, 7 months of papers and exams, but also lots of research.
That's the one, yeah.
So you kind of get a sense of what doing the research is actually like and whether it's for you.
That's the one.
And it was for you.
It was, yeah, absolutely. I really enjoyed it, and I think especially under Julian's supervision, yeah.
Yeah. So is your PhD a continuation then of the research you were doing in the honours, or did it go sideways a little bit?
It went sideways, but in a great way.
Yeah.
Yeah, so I started working on sort of a small project for my honours year, and that was really great. And doing a PhD allowed me to really open things up and look at more, allow me to be more creative and specific about what I wanted to actually research. Yeah.
So you were doing your thing rather than what you were told to do.
Oh, yeah, in as many words, I suppose.
Which is what you want, really.
Yeah,
you want some ownership of it.
Yeah, that's the one.
So you're studying photosynthesis, and I'm sure everybody kind of has heard the word photosynthesis and has a vague idea of what it is. For you, what is photosynthesis?
It's a great question. For me, photosynthesis is it's life on this planet as we know it, really, and that's something that that's a thought that I've had, or how I think about it. That's come to me only since I've been studying it, because one thing I learned is that the planet was completely devoid of oxygen in the atmosphere billions of years ago, until some organisms developed the ability to strip oxygen off water to do photosynthesis, and that's how the atmosphere was created, and that shaped life as we know it today. Yeah, in a big way. Yeah.
Yeah, the oxygen. I think I've heard someone say that oxygen was kind of like a toxic byproduct.
That's right.
Of this reaction that this organism worked out, because it had other plans for what it wanted to do the reaction for, and the oxygen was just the waste product.
Exactly, that's right. And oxygen, it's quite, as a chemist, I'm sure you're aware, is quite reactive. Yeah, likes to react with stuff. So,
yeah, I mean, that's why, for example, there's no sodium.
Yeah.
Metallic sodium in the world, because it reacts with the oxygen, and most of the iron reacts with the oxygen.
Yeah, that's right.
And stuff. Sulfur reacts with the oxygen. So, completely transformed the way that the planet was.
Yeah.
Kind of by accident.
Yeah, more or less.
But now we find it to be extremely important for us as beings who have evolved in this oxygen atmosphere.
Yeah. Yeah, it's that's the thing, right? I mean, we treat oxygen like I mean, we add like lifeblood, but we literally need it to survive. As soon as we go without oxygen as animals, we fall over and die. So it's hugely important, and not only is it important for all animals, but it's everywhere. I mean, everywhere you look, there's even in cities, there's greenness. You go out into the wilderness, it's all green, and that's all photosynthesis. So it's all around us, and I think that's really amazing.
Yeah. So your research then is not so much on what photosynthesis does, because we know that it's plants taking CO2 that we breathe out and turning it into oxygen. But you're interested in the how.
Yes. Yeah.
How does that work? And why do we care? I mean, we might come back to this, but why do we care how it works?
I think that's a great question, and because it gets me to think about, what questions am I trying to answer with my research.
Yeah.
I think it's important to know in greater and greater detail how it works because everything relies on it, and especially in a world that we as humans are changing with, the advent of climate change and creating, different conditions all around the world that influences how things grow, including plants and things. It's important that we understand all the really minute details, I think, of how it works at a biochemical level, so we can, if we need to make changes or start to engineer plants in a different way and things, if things really hit the fan, it's good to know, it's good to have all the tools in our back pocket that we can to make changes to photosynthesis or to help it do its thing, basically. And I think, and also, I mean, for the sake of pursuit of knowledge, that's what science is about, I think, for that reason as well. Yeah.
I mean, given that we're so reliant on oxygen, there isn't actually another easily accessible way to make oxygen. No. Is there?
No, not
so
really.
if for some reason all the plants gave up, we would be stuffed.
Yeah.
And so having the knowledge to have a way to produce it in another way, perhaps mimicking the way the plants do, is going to be a good thing, right?
Yeah, hugely.
And if you want to go to Mars or something, then you need to know how to do this.
Exactly. Yeah, fully. Like that's another great point. Yeah, when the conditions change of the environment of the organism that's doing photosynthesis, you might want to know this particular part of photosystem 2, which is the complex that generates oxygen. If we know what that part does in detail and we take, the organism to a different environment or something, we might know, and it starts performing differently, we might know that if we alter that part, it'll do this thing and, all that kind of thing. So, it's a big benefit to us as humans, I think, if we know how that process works in as much detail as possible.
Yeah. You mentioned Photosystem 2.
Yes.
Which is part of it. I think it's fair to say that photosynthesis, the what's going on in the leaves of the plants, is quite a complicated process.
Yes.
When you were to write out the balanced chemical equation for the reaction, it seems relatively straightforward. But what's going on in the plants is ridiculously complicated, isn't it?
That is such a good point. And it's something, I'm 2.5 years in, technically 3.5 if you count my honours year, and I haven't got everything nailed down in my head of, what actually happens from start to finish, because there are so many chemical species involved, so many things carrying electrons, and sometimes the electrons get diverted in different ways based on, what the plant or the organism wants to do. And just to add, I study cyanobacteria specifically as a model organism, and so even in that circumstance, the way that a cyanobacteria is going to deal with a certain stimulus or environment change might be different from a leaf because their photosystem 2s are slightly different. Though they're very analogous and conserved, I must say. But it's very complicated.
So there's two parts then to the process, right? You shine light on your leaf or your cyanobacterium in your case, which is a bug that can also do photosynthesis.
That's right.
And then the light energy, if I remember correctly, it's photosystem 1.
That's the one, yeah.
Which is kind of the one that generates electricity, I'm saying in quotes.
Sure, yeah.
And then there's photosystem 2, which is the part that uses that to make the oxygen.
Yeah.
Is that right?
That is right. And it's the way it's this classic story, as tale as old as time, of the way biologists name things. So Photosystem 1 was discovered first, hence the name Photosystem 1.
Yeah.
But in the order of events, chemically.
Sorry. Yes, fair enough.
Yeah, no. I mean, the way you describe it in terms of what the outcome is correct. But in terms of the order of events, you have light coming in, and in the chain of electron transfer in photosynthesis, Photosystem 2 is the first one that does anything. Light comes in, it excites an electron on a chlorophyll molecule. I'm sure people have heard of chlorophyll before.
So that's what makes them green.
Yeah, exactly. Right. Yeah, all these pigments, these things that are trying to absorb light in the visible spectrum. That's why they look so vibrant to us, is because we also see the visible spectrum of light. And so that electron gets excited, and as it does so, that electron's going to go down the chain and it'll wind up at a bunch of other complexes and Photosystem 1, like you said. But that electron needs to be replaced by something, and in this case, it's water. And that's, for me, one of the most amazing parts about photosynthesis, is that it's able to strip electrons off of water, which is an incredibly chemically intense process.
Yeah, because water's pretty happy as it is.
Oh, yeah, big time. Yeah,
as you can tell from how much water there is around the place.
Exactly,
it's a very stable molecule.
Yeah.
So doing chemistry on it, and this is why, of course, it's important to understand how photosynthesis does it, because for us, that's really hard.
Mm-hmm.
Yeah,
big time. I mean, that's the thing, right? Like, there's so many ways that we are trying to mimic how Photosystem 2 does pull electrons and oxygen out of water. And so far, we can't come nearly as close as the efficiency of Photosystem 2. I mean, I forget the exact percentage, but it's in the tens of percents, like 60, 70, 80% efficient, I think, in terms of how much light energy goes in versus how much oxygen you pull out and things like that. It's very, very impressive that life has managed to get to that efficiency level.
So, very complicated process.
Mm-hmm.
Lots of proteins involved, as well as these chlorophylls and stuff.
Yeah.
And from reading the notes that you sent through, your particular interest for your research is on one of these proteins and exactly how that plays its role in the overall thing.
Yeah, that's right. So you're completely correct. Photosystem 2 and all of the other complexes involved, but specifically in Photosystem 2's case, it's made up of a bunch of different proteins that come together to perform this function of splitting water and producing oxygen. And the protein I'm focusing on is called the D1 protein. The gene that encodes for this protein is called PSBA. And so what I'm looking at is the different a few different amino acids in the D1 protein that are having certain roles, or that may be or may not be having certain roles in photosynthesis. And an amino acid is basically the building block of a protein. A protein is just a really, really long molecule that folds into a certain way. And it folds in a specific way given what the sequence of specific amino acids are in the protein. So I think the D1 protein is maybe about 300 amino acids in length. It's quite big, relatively speaking, for a specific protein. And it has life in general, biology has about 20 different amino acids to choose from. So if you think about the number of sort of sequences you can make, immediately the possibilities open up for things to fold in specific ways. And so the D1 protein folds in a specific way to have this specific function. Yeah.
And the three-dimensional shape that the protein ends up in after it does the folding is crucial to the way that it does its thing.
Absolutely.
Just as a segue, the Nobel Prize for Chemistry has been announced just in the last week, and it's been given to people who have, and I was unaware, I'm completely not keeping up. Yeah, right. People who have developed ways to actually use computers to predict how this folding goes.
You're absolutely right.
When I was an undergrad biochemistry student, this was science fiction.
Yeah.
And then I thought we were still at the point where, in theory, machine learning and AI could do this.
Mm-hmm.
Little did I know it's already happening.
Yeah.
So is this something that has been of use to you? Because the traditional way of working out the structure was to use something called X-ray crystallography, which is an actual experimental method rather than a computational method.
That's right.
So your protein that you're interested in, do we know what the actual three-dimensional structure is, one way or another?
We do, and we do via actual experimental methods like you mentioned. And I think that's probably because, this is such an important protein and does such important things that it's been studied, well before the advent of machine learning, so we know what it looks like. But you're right. I mean, it's absolutely mind-blowing that that AI now, machine learning, is now able to just predict what a protein is going to look like in 3D space. Tell you what, I mean, even when I was in my undergrad learning about protein folding and Ramachandran plots and all this sort of thing, it was told to us, and it made sense how monumental of a task it would be to figure out how something like that would fold, given all the different potential energy wells things could fall into, or all the different ways things can just interact in 3D space.
Yeah.
Now, in the space of 5-6 years, AI has just come a long way. But yeah, given for my case, since I know what the protein looks like already, I'm able to use software on my computer just at home to load up what that structure looks like, render it in 3D space, and actually go in and have a look at the different amino acids and where they are relative to the chemicals involved, like plastoquinone B, I've mentioned, or at least in notes to you. But for those at home, plastoquinone B is a quinone molecule that carries 2 electrons and 2 protons out of photosystem 2 and down the chain, and that's sort of how that all gets down to photosystem 1 and things like that. And so what I'm looking at is how different amino acids, specifically in the region where QB comes in to accept those electrons and protons, how those amino acids might influence QB's binding, how it accepts the electrons, and the stability of the molecule when it sits in photosystem 2.
I was thinking about this when I was reading your notes and thinking about analogies of what you're trying to do, because what you're trying to do is you're trying to change an amino acid for something else and see what happens.
Mm-hmm.
And where I got to was it's kind of like trying to work out how a car motor works,
right?
By just going in and pulling out a piece,
and
seeing if it still goes.
Yeah,
and by doing so iteratively, you can work out what the function of all the different bits and how important they are to the overall running of the engine.
Exactly.
And so this is what you're kind of trying to do with the components, which for you are the amino acids.
That's right. Yeah, that's a great way to think about it. And I can, it's like that, but already maybe having some kind of understanding of the working of an internal combustion engine, but at a very rudimentary kind of level. Like I can understand, for example, how an amino acid with a very non-polar hydrophobic side chain might interact with an oxygen molecule on QB, for example. Probably not much is going to happen. It might just, float around. Whereas if I take that side chain and introduce a nitrogen and an oxygen on the end of it, suddenly that oxygen on the end of QB is probably not going to be too happy. It's going to be, repelled by both of them being quite electronegative. That's an example of something that I've done.
Yep.
Is I've taken an amino acid side chain and introduced some electronegative groups on the end of it, and I'm already seeing in some experiments I've been doing just this past week that photosystem 2 is not too happy, and in fact it's having a really, really hard time seemingly getting QB into that site, accepting those electrons and pulling them out.
Right.
Not only that, we're also seeing some very interesting energy transfer phenomena where photosystem 2 seems to be receiving a lot more energy from surrounding photosensors, we think, than usual, which is strange, and this isn't really known in the literature, that a malfunctioning photosystem 2 might have this effect. So
you're short-circuiting it somehow.
Yeah, something like that. I mean, it's almost like the cell is trying to remedy what I've done, because I've really buggered things up, I think, with this mutation, and so it's trying to fix this problem. But that's the amazing thing about biochemistry, is that if we create issues, we can also learn so much else about the cell because it tries to fix those issues, and it might create a response that we can study that we weren't even aware that existed. So yeah, it's really, really, really incredible and fascinating research. Yeah.
And to be clear, even though you were saying that you could sit at home in your bedroom on your computer and actually model what's going on, you're actually making new versions of these proteins in the lab, aren't you?
Yeah, that's right. And I feel very lucky to be in that position, in the sense that the organism that I use to do this is super cooperative. That's the word I'm looking for.
Right.
So people at home may have heard of things like CRISPR-Cas9 or CRISPR gene editing and things like this.
Yep.
That's quite a popular term in pop culture now. And the reason why it's so popular is because traditionally, I think at least in eukaryotic cells, so those are cells in plants and in humans and things like this that are complex, that have their DNA in a nucleus. Editing that DNA is no mean feat. Yeah. Or it is a mean feat. It's hard to do.
It's very hard to do.
Very difficult. Whereas with the cyanobacteria that I'm working on, you can just take a piece of DNA that I can make, in a PCR machine that's polymerase chain reaction. I can just amplify a sequence of DNA, put it in a test tube with the bacteria, and it will just take it and put it into its genome and be happy.
Start making proteins.
Exactly. And start making proteins the way I want it to.
Yeah.
Yeah. And I can use, very sort of simple tools like antibiotic resistance selection to select for the ones that take up the genetic material that I give it, such that I can grow now, so I can grow bacteria with copies of Photosystem 2 with these mutated amino acids. I can grow mutant bacteria where Photosystem 1 is completely knocked out. I can truncate the 2 core proteins of Photosystem 2 and just knock them completely out. And it's very interesting. When you do that, you get this, what is normally a very green kind of vibrant cyanobacteria, and it goes bright blue, because you've just taken out Photosystem 1, which holds a lot of those chlorophyll molecules, and so you really impact the way that it appears to us visually.
Yeah,
those pigments.
You're changing the composition of the whole machinery.
Yeah, exactly.
Yeah. So you've already indicated that you're finding out some interesting things, and you're almost finished. So if you had to summarise what you've learned in your almost 3 years now, in 1 minute.
Give it a go. I think I've learned that life has so much more complexity to it than I ever thought it did or could. When I came into my research, into my lab, I had the same reaction that I think a lot of people do when I tell them what I researched, that, I thought we figured out photosynthesis a long time ago. But it turns out that, there's so much more to it than we ever realised.
In fact, there was a Nobel Prize given for, I'm going to embarrass myself with the names now, but it was, in the 80s? For the guys who first worked out Photosystem 1 and Photosystem 2, that they were things, maybe.
Yeah.
I remember there being a poster on the wall in the lab when I was an undergrad for that.
I should probably know their names, to be honest.
That's a long time ago. Introduction for your thesis.
Yeah.
Not yet.
Yeah.
But a long time ago. And yet still, 30, 40 years later.
Yeah, and I think that it puts into perspective how small life gets. It goes right down to the atomic interactions, and in fact, it even goes further, smaller than that. It goes to like quantum interactions of electrons.
Yeah,
this thing can happen in Photosystem 2, where if you have an electron held at an excited state in Photosystem 2 for too long, it can flip its spin. When it does that, it can no longer relax down to what's called its ground state. And when that happens, it can interact with molecular oxygen and turn that into what's called singular oxygen, and that can be incredibly, disastrous. And so
free radicals and such.
Yeah, exactly.
Yeah.
So I think the biggest thing I've learned is that there is so much more to learn about life than I ever realized. And I think that's a really beautiful thing, because it means as humans we'll have, we still have so many things to learn and so much time to learn it. And I think it's really amazing, and it's motivating, it's engaging, it gets me to really engage with science so much more now.
And even though the AIs and stuff can help and are helping, they still can't go and make you the molecules. Make you the proteins.
Exactly.
And that's what you actually need if this is going to be of use to us when we go to Mars or whatever.
That's the thing. You need you need the human element. You need someone who's going to, go and physically do the things and have it's the versatility of the human mind that can both just think maybe like, an AI could, but also have that sort of creative spark, the imagination, and then the actual drive to, get your boots on the ground and actually go and do the thing. Yeah.
Cool. Well, we're out of time. Thank you, Toby.
No worries. It's been
fascinating
a pleasure, Dave.
stuff.
Yeah.
All the best for what's left of the thesis and whatever's next for you.
Thank you very much.
Thanks for listening, everyone. You've been listening to the Science Notes podcast. brought to you by the Division of Sciences at the University of Otago. To listen again, or to find other episodes in this series, go to Otago Access Radio's website, oar.org.nz, or to your favourite podcast platform.
This podcast was produced by OAR FM Dunedin with support from New Zealand On Air.